Welcome to The Long Run. This is a podcast for biotech adventurers. I'm your host, Luke Timmerman. Today's guest is Barbara Weber. She's the CEO of Cambridge, Massachusetts-based Tango Therapeutics. Ever hear the saying, "It takes two to tango"? That's where this startup draws its inspiration. It's seeking to discover and develop synthetic lethal cancer drugs. What does that mean? These are intended to target two molecular vulnerabilities of a cancer cell, not just one, like the first wave of targeted therapeutics did 20 years ago. The concept of synthetic lethal is a hot one today in cancer biology. Venture capital has been flowing for a couple years now to companies like Tango, Repair Therapeutics, Idea Biosciences, I hope I pronounced that one right, and Cytier Therapeutics, all looking to stake their claims. Tango, like many startups, is seizing upon a confluence of advances in technology that makes synthetic lethal cancer drug discovery more practical now than ever before. There's fast, cheap DNA sequencing, fast, cheap CRISPR gene editing, and improving bioinformatics that help narrow down the possibilities. This company, founded in 2014, is still at quite an early stage. It has no drug candidates in the clinic, and it will be years before it gets there, if ever. I've written about Tango before and invited Barbara Weber to speak about Tango last spring at my Boston Cancer Summit, the charity fundraising event for my Everest Climb to Fight Cancer campaign to support research at the Fred Hutch. Curious to hear more, I made a note to invite her on the podcast after getting home from the mountain. And it so happens she's been busy. Today, Tango is announcing its first big partnership. Gilead Sciences is paying $50 million up front to get the option to license 5 new targets that come from Tango's discovery platform. Tango retains 100% ownership of its lead programs. It's not saying what they are. But Tango didn't have to mortgage away its future just to get cash to keep the doors open a few more years. And this deal is yet another sign of the times, in that startups with promising technology in today's boom are bargaining from a position of strength, and the big wealthy companies are under pressure to fill up their pipelines with things that might excite their investors. As for Weber personally, she has the kind of resume you'd expect of someone serving as the dean of genetics at some distinguished university. She's an MD by training, did a residency at Yale, fellowship at Dana-Farber, became a professor of genetics at Michigan and Penn. Then she went to the other side of the fence in R&D leadership roles at big pharma stalwarts GSK and Novartis. She ended up in the orbit of Third Rock Ventures, the Boston and San Francisco-based venture firm that starts companies focused on emerging areas of biology. She worked for a while on another Third Rock startup, the personalized neoantigen cancer vaccine developer Neon Therapeutics, before being drawn in to run Tango full-time. Hearing her describe her career arc, Barbara sounds like she's gotten herself into the right kind of situation where she and her team can execute on something she's long wanted to do. Okay, now before we dive in, a few plugs. Life Science Cares is an exciting new philanthropic outlet for biotech professionals. I've personally contributed because I believe in its mission of fostering community volunteerism in biotech. Never heard of it? Here's the gist. It's a collective effort of the life science industry to end poverty in the greater Boston area. Now working with more than 150 companies, Life Science Cares harnesses the industry's human and financial resources to support service organizations that are doing the best work in the areas of basic human survival, education, and economic sustainability. To learn more about how your organization can become a member or how you can volunteer, visit www.lifesciencecares.org. And if you enjoy listening to these in-depth interviews, you'll love reading Timmerman Report. You can subscribe for $149 a year per person. Go to timmermanreport.com/subscribe. Lastly, are you planning a conference, a team-building event, or a leadership retreat? I've developed a presentation based on my successful Mount Everest summit expedition. My Everest talks feature gorgeous photos from the world's highest mountain, along with lessons on leadership, teamwork, and what it takes to overcome adversity to achieve the big goals in life. Interested? Ask me about an Everest talk at luke.timmerman@protonmail.com. Now join me and Barbara Weber for The Long Run. Welcome, Barbara. Thanks for joining me on the show.
Thanks for having me, Luke.
So I'm really excited to have you on the show here today, Barbara. You were one of the featured speakers at the Boston Cancer Summit event that I organized in the spring to raise money for the Everest Climb to Fight Cancer at Fred Hutch. I want to thank you for that. Your story is still, it's early, and I'm curious to see how it's developed over the past, say, 6 months. But before we get into some of the meaty stuff that you're doing today, can you just tell me a little bit about yourself and where do you come from? Where were you born and raised?
Sure. Oh, wow. Well, that's way back. So I actually was born in Palo Alto, grew up mostly on the West Coast, but then and went to college and medical school at the University of Washington in Seattle, but moved back east to do my internship and residency at Yale, and then my medical oncology training at Dana-Farber, and have been on the East Coast pretty much since then.
Okay, well, let's go back just a bit here. You say born and raised in Palo Alto near Stanford. What did your mom and dad do for a living?
Oh, my father was a stockbroker, but I think actually my family ended up being out there because my grandfather was a physicist and was at Stanford, and that's how my parents ended up there.
So what about your mom?
My mom didn't really work when I was growing up, but she was a dietitian and went back to work after we were out of the house. Yeah. So.
And siblings?
I have 2 sisters, one of whom is a DVM PhD. So she's a veterinarian who's an academic veterinarian at Washington State in Washington, and at the vet school there, and another sister who lives in Houston, Texas.
I'm based in Seattle, so I'm familiar with Washington State University, Pullman, Eastern Washington. It's a very good veterinary school, by the way. Okay, so were you the oldest of the 3?
Yeah.
So you did allude to some of these stops at illustrious institutions that came later. But how did you, even as a kid, how did you get interested in science or medicine?
That's an interesting question. Yeah, and I think it's funny that you asked me about my mother being a dietitian, because I think in some ways that was the link. And I also really enjoyed books as a kid about people, healthcare providers, I guess we'd call them now, who did important things for people. So, kids with cerebral palsy who physical therapists changed their life, things like that. I loved to read those books when I was a kid. And so I started thinking about physical therapist training and school. And then the more I sort of got interested in science, and maybe even at that point sort of understood my own personality, I thought, I really need to be a doctor and go to medical school. And so that was the original link. But I also think that I've always been interested in genetics and what genetics could mean for thinking about both science and taking care of patients. So that was also another piece of it for me from the very early parts of my thinking about what I would do with my life.
Now, we're in Palo Alto. Were you in public schools?
I did. Yeah, I went to public school.
Okay, so I would imagine.
So
a lot of
was very different. Palo Alto was not like what Palo Alto is like now.
Well, I didn't want to be rude and try to date you here, but yeah. So it is near Stanford, and this whole milieu. I would imagine there's a lot of smart people around. Kids of professors going to your school. Was it a competitive kind of place?
Well, it was. Now I've abbreviated some of my history, right? If you want to really get down to the details, actually, my father decided that he could make his fortune being a stockbroker in Alaska while the pipeline was being built. So I actually went to high school in Anchorage, Alaska. And that is in some part how I ended up at the University of Washington Medical School through the WWAMI program that you're probably familiar with.
Oh, yes. Okay, so for those who are not, this is the WWAMI. Not W. It's Washington. It's the northwestern states that only have one medical school. At that time, University of Washington, to serve, I think, 5 states.
Yeah, when I went, it was 4. It was Washington, Alaska, Montana, and Idaho. And I think there's another one that's been added.
Well, I think they added Wyoming. That's the other W.
I think that's the other W. That's right.
Okay, so you're growing up and going to school in Alaska, and then coming to University of Washington. Okay, go Huskies. So what did you study there?
I was a chemistry major.
So how did that captivate your interest in and lead toward
Oh my
a medical
goodness,
school
Luke,
career?
you're sending me so far back. It's interesting how individual people, I haven't even thought of this for a while, but individual people can influence you. I had what I thought was a really great chemistry teacher in high school, and I just thought it was very interesting. So I decided I would be a chemistry major, and I liked it and stuck with it. So I took the pre-med courses along with it, but I really enjoyed the chemistry piece of it, organic chemistry, physical chemistry.
That's really interesting. And so I actually, this is a theme of mine. I'm curious about people who inspire others and encourage people. And everybody has somebody like that. And for you, it was that chemistry teacher, and that's great.
It was also, there's another person, again, you may know him, or I think his son now, George Stamatonopoulos, who was the genetics professor my first year of medical school, that also got me really interested in medical genetics. So there are, at each point in my career, I can tell you, that there was often one or two people who really inspired me and sort of by being who they were and how they influenced me, very important in decisions I made subsequently.
Well, I don't want to belabor it too much on the University of Washington, but in those days, in genetics, it was already emerging as a powerhouse. Arnold Matulsky was there. George Stam, as you mentioned, his son, John Stamatoyannopoulos, is still here, and he's running an institute that's supported by GSK. There's a lot of great genetics history here. And it sounds like you caught the bug.
I didn't know that when I just showed up for, the medical genetics seminar. And in fact, I guess it was my second year of medical school. I was just blown away by the sky and thought, this is amazing, right? And of course, it turns out then all of that was also around.
Okay, but then you go on and you become a doctor. And I'm looking at your career arc, and this is really quite... It's like you've been to so many illustrious institutions. You did an internal medicine residency at Yale, oncology fellowship at Dana-Farber, later a professor of medicine at Penn, stop at GSK and Novartis. Wow. You've really done a lot. Could you just talk at a couple of those important turning points? Which ones were important to lead you to where you are today?
Well, that's a really interesting question, and I think they were all really important, at least in the sense of the fact that I spent a long time in academics. I had a significant history in big pharma, and now I'm in biotech. And I think that, at least for me, I'm able to do what I do now, my job as the CEO of Tango, for me, what feels like a much better way than if I hadn't had all those different experiences. More often people come with one of those experiences, or sometimes two, but usually not all three. So for me, they've all been equally influential, and I've had some really amazing experiences and learned tremendously from each one of them. I could pick out maybe one or two things from each, but I have to say that I feel so fortunate to have had all those different experiences.
In terms of what you're doing today with synthetic lethal cancer drug discovery, it's clearly the cancer part and the genetics part are both very relevant in your background to what you're doing today. So maybe can you just tell me a little bit about what did you gain during that? It sounds like oncology fellowship at Dana-Farber would have been one kind of formative experience. What did you learn there?
I would say it was actually at the next step, which is when I went to the University of Michigan, which was my first faculty appointment after being a fellow at Dana-Farber. And it was an amazing time there at Michigan. Jeff Leiden was there, and Craig Thompson was there, David Ginsburg. There was just, Francis Collins. There was this amazing group of people that had been assembled at the University of Michigan in the School of Medicine. And it was really there that I got introduced to Francis Collins and started working with him on the isolation of BRCA1. That was a major turning point for me. And it actually really was the beginning of thinking about what I do now, because we thought at the time, this will be great. We'll clone this gene and we'll be able to actually develop a drug, or somebody will be able to develop a drug that, addresses these breast cancers in these women and maybe prevents it. But of course, it's a tumor suppressor gene, and it's gone or it's inactivated. And so therefore, by definition, not druggable. And fast forward now, 15 years later, 20 years later, that CRISPR as a technology now enables us to use that genetics concept of synthetic lethality to find ways to target tumors that have specific tumor suppressor gene loss. So going really all the way back to the University of Michigan and my role in the BRCA1 and 2 projects, which were heavily influenced, by my working with Francis, that probably more than anything drove what I've been really focused on since then.
This would have been the 80s, and a whole world was opening up with oncogenes and tumor suppressor genes, even before we had the genome.
Right. So Mary Claire King identified the locus for BRCA1 on chromosome 17, I think, in 1989. And that's when this whole piece started.
Okay. Now, we're going to get real quick into the synthetic lethality concept. But before that, you did a couple stops in Big Pharma at GSK and Novartis. What was sort of the thing that drew you there?
Well, what drew me from academics into Big Pharma in the first place was that, I'd been working on BRCA1 and 2 for a number of years, had really done a lot with characterizing mutations and built a big translational program there for women and their families, and had done really a lot of work in that space, as well as the cloning of BRCA2 with the Myriad group. But after some time, it just it was sort of weighing on me that we really weren't being able to do anything about it except talk to people about their risk. And at the same time, Novartis had developed Gleevec. Brian Drucker presented the data from Gleevec at that AACR that year. And then after that, I think it was, a number of people, but let's say David Tuvison and George Dimitri, who proposed that Gleevec should work in KIT, in GIST, because it hits KIT. These things were all happening for me at the same time. And I said, I am kind of tired of having the output of what I do be papers on my CV. I really want to think about how I could stop having just be the first paragraph of every grant say this might be important for patients in this way, to really learning how to and being part of making drugs that really were directly important for patients. And that's actually what drove me. I had a big grant from GSK, and I was working with them on how to use Array CGH at that time, another genomic platform, to say, how do you find new cancer targets? And they offered me a job, and I said, yeah, sure.
Okay, but I mean, at this time, you're a tenured professor, right?
Yeah, it was very shocking to a lot of people. I had lots of grants, and I was a full professor and a big group, and I said, yeah, no, this is what I want to do. I want to take everything I've done now and really start to think how we could turn it into something that matters for patients. And when I went to GSK, I really learned a lot about how to make drugs, but I remained sort of frustrated about how genetically targeted drugs were being developed. People were still developing them as if they were chemotherapy. And I had, because of my genetics background, I really had this, idea in my mind that we could dramatically change how cancer-targeted drug development was being done by selecting patients correctly from the very beginning of phase 1. And at the time, now it was 2009, David Epstein was still the head of Novartis Oncology, and he offered me a job to come run translational medicine for Novartis Oncology, which is the whole middle part of the pipeline. So everything from candidate selection of the drugs in the oncology portfolio up to phase 3, and basically carte blanche to change things the way I thought they needed to be done. And that's why I went to Novartis, and that's exactly what happened. And he's another person in my life who was just extremely influential to me. But it was the Novartis portfolio and resources and then Hervé Oppenau's leadership that allowed us then to really change how we were doing clinical trials and move from all comers in phase 1, thinking that you could slowly narrow that down over phase 2 and phase 3, to what we really did, which was, first of all, eliminate phase 2 altogether and select patients genetically from the very first patient on the first dose. And I actually have the data from this. What that did was take the time from first patient dosed to clinical POC from a median at that time of 4.5 years to 7 months.
Yeah. These are the years when sequencing is getting a lot cheaper and better and faster, so that enables some of this to happen. What years were you there at Novartis?
I was there from 2009 until 2015. And during that time, I think for me, a real highlight for me was that using that approach, we developed ceritinib, which is Novartis's ALK inhibitor. We selected patients for ALK translocation from the very first dose. We saw our first response in the second patient who was treated, who was a 24-year-old college student. And we registered that drug, with the help, obviously, of Alessandro Riva and the Full Development Organization, who did the heavy lifting on the registration within 3 years of the first patient dosed.
This is part of the promise of, I guess, what we call precision medicine now, in that it is supposed to, for the pharmaceutical industry, deliver this promise of really utmost importance to reduce the time, the risk of failure, the cost, everything, to make things more efficient in R&D.
And it absolutely does that. When you have the right target and you select patients from the beginning, it absolutely does it. You can go back and look at what Peter Leibowitz did with the MEK and the RAF inhibitor and the combination at GSK. You can look at ceritinib. You can look at the alpha-selective PI3 kinase inhibitor Novartis just registered. You can go all the way back to the first company that did it, which was Genentech with Herceptin. It really works every single time when you've got the right target and you select the patients.
Okay, so 2015, you come to another juncture. Now, I want to make sure I have my dates right. Was this around the time when the PARP story kind of cracked open this whole aha moment for synthetic lethal cancer drugs?
Well, from my perspective, the PARP story cracked it open way, way before that, because Alan Ashworth, who's a friend and longtime collaborator of mine, is the person who discovered the BRCA1/2 PARP interaction. That was in 2005. And Alan's the founder of Tango. I think it was back then where Alan started working with a company called Kudos that was ultimately bought by AZ with a drug that ultimately became olaparib, that became absolutely clear this is how to think about tumor suppressor genes. And actually, it's a little bit of history. Alan and I, way back in 2005, wrote up a small business plan and pitched it to a VC company that I won't name, but it wasn't Third Rock. They didn't exist then. And suggested that we could build a company based on synthetic lethal screening and find targets for tumor suppressor genes. And the feedback we got was absolutely right. You can't do this. It's not technically possible. And that is true, because back then we didn't know it quite yet, but shRNA approaches just give you way too many false positives for that approach to be done at scale. CRISPR is what changed that. And so literally, what we do at Tango is not that different to what Alan and I proposed literally in 2005, but we can actually make it work technically now because of being able to use CRISPR libraries.
Well, and this is an important point I want to ask you about in a minute, but I guess for those who are not that familiar with even the concept of synthetic lethal, basically you're looking for two vulnerabilities in the tumor genetically, not just one. And can you elaborate?
Sure. The concept was actually discovered a long time ago in the 1930s by fruit fly geneticists, and what they recognized was that there were flies that had certain phenotypes, which it's unbelievable to talk about this. It was even before DNA was really discovered. And they didn't know that they were DNA mutations. But these flies had phenotypes that you could see under the microscope. And they knew that, for example, flies with one eye phenotype, like glass eyes, and another eye phenotype called bar eyes, those two flies by themselves were fine and could reproduce. But if you mated those two flies, all the offspring were dead. But there were other combinations like wingless flies and glass eye flies that when you mated them, nothing happened. So that's the concept of synthetic lethality. There are pairs of mutations that can be tolerated together, and there are other pairs of mutations that can't be tolerated together. But all of them are fine by themselves. And that's actually exactly the concept that we use in cancer to say, the cancer has one mutation. How do we find the drug target that is, quote, synthetic lethal with that drug target, that the cancer can't tolerate, but the normal cells can tolerate because they don't have that mutation? that's exactly PARP, right? So normal cells, not much happens when you treat them with a PARP inhibitor. Cells with the BRCA1 mutation or loss of BRCA1 will die when you give them a PARP inhibitor because they have now not just the PARP and not just the BRCA1, but they have them together.
Now, in the mid-2000s, you mentioned you had the bug for an idea of a synthetic lethal company. But at that same time, the PARP inhibitor drug candidates were moving along. People were excited about them just because of PARP. I don't think they were really thinking about the BRCA1-2 connection at that time. But a lot of those drugs failed. Was this a matter of sort of looking through the subset analyses and finding, the drug is failing in the all-comers population, but boy, it looks really encouraging in this subpopulation with BRCA1-2 mutations?
Yeah, that could have worked, but it actually isn't what happened. Alan actually hypothesized that BRCA1 or 2 and PARP should be synthetic lethal. And it goes back again to a basic genetic concept, which is that these synthetic lethal pairs are often two different genes in the same functional pathway. So BRCA1 is a DNA damage repair pathway gene. PARP is a DNA damage repair pathway gene. And Alan thought, let's see if any of these drugs that are out there that are available that are DNA damage repair pathway inhibitors are synthetic lethal with BRCA1. And he got the PARP inhibitor from Kudos, and he showed that and published that in I think it's a Nature paper from 2005. After that, AZ got interested in it, and they bought it for that purpose. But, as sometimes can happen, there was a very circuitous kind of route to getting there and getting everybody behind selecting patients and all that kind of stuff. So, yes, you're right. The drugs were being developed as sort of non-specific chemotherapeutic agents, if you will, although the target was known. But the synthetic lethal effect for olaparib was known from 2005.
I see. But around, just to get these timelines right again, around, that 2015 timeframe, a number of things are happening. CRISPR, which you alluded to, but there was also one of these late stage clinical readouts. I don't know if it was with AZ or there were other companies like Clovis and Tesaro that were also zeroing in on this PARP BRCA1-2 connection. And it became validated
Absolutely.
clinically.
And it was a happy coincidence for us because we actually already had the Series A funding for Tango, and we were getting ready for the public launch a few months later, which was in March of 2016. And it happened almost coincident with that first study, or that study that I think was niraparib, now that I think about it, that was the really dramatic effect in ovarian cancer.
After, it sounds like a really interesting, good run at Novartis, 6 years, lots of resources, lots of cancer drugs in the pipeline, you decide to get into the startup world. You joined Third Rock. What was your mandate then?
Well, like all things, there's a story, which is that when I went to Novartis, the other thing that happened was that Francis introduced me to Bob Tepper. So now it's like late 2008, and they're starting Third Rock. And I met with Bob, and he said, we're just starting this new company, Third Rock, and we're going to do this cool stuff and creating companies, and we're starting these two new companies, and they both need a CSO. Would you be interested in either one of them? One of them is going to be called Agios, and the other one's going to be called Constellation. So at the same time, I was being offered the Novartis job, and I said, I'm condensing a lot, of course, here, but I said to Bob and Mark Levin that I really thought what they were doing was so cool. They were, going after important problems with the best people in the world, and they were putting a lot of resources behind it. I was going to take the job at Novartis because it was sort of, as I was explaining to you a minute ago, it was what I really wanted to do then, and it was the right place to do it. And I said, these companies are far from the clinic, but I want my next job to be at Third Rock. That was in 2008. And I stayed in contact always with them. In fact, I'm a huge Foundation Medicine fan for probably obvious reasons. And it was Mike Polini and I that did the Novartis Foundation Medicine deal, which was the first pharma deal for Foundation. So, it was really through those connections that when it was clear it was time for me to leave Novartis, I called Bob and said, what do you think? And he said, yeah, come on over. And to be honest, I didn't know anything about company building or venture capital, but, I knew what I wanted to do, and they felt like the people I wanted to do it with, and it was a great choice, and it was a great opportunity.
Well, it is a small world there in Boston. You mentioned Foundation Medicine. That's part of the Third Rock portfolio. Several Third Rock members on their board, et cetera. You already know these people. So you had a pretty good sense of who they are and what they do, but maybe not company building, like what that was all about. That was new. But you went in, was it like a venture partner or entrepreneur in residence kind of role where you sort of look around for a while to see what it is that you will devote yourself to full time?
Yeah, I started out as an entrepreneur in residence, and after a year they said, and I said, yeah, I'd love to be a venture partner. I want to do this. This is great. This is really fun and right in my wheelhouse. And going back to what we talked about at the beginning, having the academic background and the drug development experience was kind of perfect for that. And I'm sort of an organizer, builder kind of person anyway. So I, literally within a month after I joined, Bob Tepper and Carrie Pfeffer were launching Neon, and Carrie is the interim CEO and Bob is the interim head of R&D. And they said, would you be willing to be the interim CMO? And I said, I don't really know anything about cancer vaccines or immunology. But wow, what a chance to see how you launch a company, right? So I was the interim CMO for Neon from the Series A to the Series B when we brought in Hugh O'Dowd and Richard Gaynor in those positions. And I also worked closely with Alexis building what became Relay from when it started as an idea up until its launch. And so in the background now, I'm getting started with Tango, which I built myself at Third Rock with Alexis's help, and then Carrie coming in with me as an interim C-level person here when we launched it. But even when we launched Tango, I was saying, I don't want to be a CEO. I want to stay as a venture partner at Third Rock. I'm going to replace myself as the CEO of Tango. I'm going to go back to Third Rock and make some more companies. But what happened is that, this was been my baby since like 2005. I've been thinking about it. The team, because I started this at Third Rock myself, is all people that I brought in. The science was just turning out to be just as amazing as I hoped that it would be. And every time Alexis said to me, you've got to start interviewing CEO candidates, I'd say yeah, maybe next month I'll do that because I don't really want to give this job to somebody else. So finally last summer, Alexis said, look, you got to hire a new CEO or you got to take the job yourself. And so I said, okay, great, I'm doing it. So now I'm a very part-time venture partner at Third Rock and the permanent full-time CEO of Tango, and it's been a great choice for me.
Life Science Cares is an exciting new philanthropic outlet for biotech professionals. Its mission is to end poverty in the greater Boston area. To learn more about how your organization can become a member or how you can volunteer, visit www.lifesciencecares.org. And is your company interested in raising its profile? Cut through the noise. Reach biotech leaders who listen to this show and give it their undivided attention. This is an opportunity to seize. Sponsor the Long Run Podcast. Ping me at luke.timmerman@protonmail.com. So Tango, and the name is one of these that's quite instantly appropriate, right? It takes two to tango. It takes two genes to really neutralize the protein, the tumor. Okay, now. But you mentioned CRISPR. I want to come back to this because this is an important point. I think a lot of people, when they hear that term, they think of CRISPR drug companies. Companies that are fundamentally using that to clip out some disease gene or maybe knock in something. But actually, CRISPR is being used much, much more widely across drug discovery platforms for lots of different purposes. At companies that don't really advertise themselves as CRISPR companies. So how do you use it to enable your discovery engine? Yeah,
and exactly what you said is right. We use it for research purposes, not for therapeutic purposes. That's really the important distinction, right? So Editas, Intellia, CRISPR, those companies are actually proposing to treat patients with CRISPR as a drug. We use CRISPR as a way of finding novel cancer targets. And then we use sort of more conventional small molecule drug discovery approaches to make the molecules that the patients will be treated with. So for us, CRISPR is a tool. It's a genome scale tool that we can use to find the targets.
And there are a lot of companies that are trying to put CRISPR to work in drug discovery. I mean, even at my event in the spring, we had KSQ Therapeutics, David Meeker, who you know. And the story is similar, that you can screen combinations at high throughput.
Yeah.
Can you explain a little bit more what you mean by that? What kind of questions can you ask, and what kind of speed and volume are we talking about?
Sure. So, going back to what Tango is about, it's about finding pairs of mutation drug combinations or even drug-drug combinations in specific genetic contexts that will kill cancer cells and not harm normal cells. We also feel strongly, and the data support this, I think, that these synthetic lethal pairs are context dependent. So, for example, something that's a synthetic lethal pair in gastric cancer may not be the same synthetic lethal pair in lung cancer. So what we do is two things. One, we identify a patient population that we want to address, that high unmet need. Now, you could argue, and I think it's right, that essentially almost all cancer is still a very high unmet need. But let's say patients now who don't have good targeted therapies and are not really responding to immunotherapy. So let's say all of GI cancers, right? Gastric cancer, colon cancer, all of those cancers are still places where the therapeutic options are pretty limited. Then we say, okay, what do the genetics of those cancers look like, and subdivide them? So there's a subset of gastric cancer that have ARID1A mutations or ARID1A gene function loss. That's maybe 30 or 40% of gastric cancer. So then we'll look for, say, 5 or 6 or 7 gastric cancer cell lines that have ARID1A loss and a matched number that don't. That's what we start with. What we use to look for targets then is a druggable genome CRISPR library. It's about 5,000 genes that by making a CRISPR library out of them and infecting in pools those cells of the 5 or 6 or 10 different cell lines, we can tell through sequencing techniques which of those 5,000 genes are potential synthetic lethal pairs with ARID1A in that context. And those pairs, that other half of that pair in those experiments, are the potential drug targets. Then we go through a series, of filters of saying which of those are potentially the most promising drug targets. We do standard, biologic validation for those targets, and then we would take that target into drug discovery.
You use that phrase, context dependent, and I think that's an important one because I think people have this idea that, okay, maybe you've got gene mutation A here and gene mutation B here, and the organ of origin may not be that important, but what's important is the mutational status of A and B. But what I think I'm hearing you say is that actually that context is important. So maybe gene A and B are misregulated somehow or mutated in gastric cancer, but it won't just be translatable automatically to other cancers that look like that molecularly in other tissues.
That's exactly right. And there's examples of that already, right? Take, for example, RAF inhibitors work really well in RAF mutant melanoma, BRAF mutant melanoma. Really don't work at all in BRAF mutant colon cancer. The difference is probably both the lineage program, which has, effects that we are just beginning to understand, and also that the mutation spectrum is different. The other commonly mutated genes in melanoma and in colon cancer are very different. So you end up with that exact setting. BRAF inhibitors are context dependent in their efficacy, right? It's exactly the same thing. And I think it's also important, I'm glad you brought this up because to me it's just fundamentally important, is that with the Broad Project Achilles and Novartis Project Drive, the idea behind them, which made sense, at the time, and still they're very valuable data sets, is let's talk about the Broad Project Achilles. Let's assemble 1,000 cancer cell lines and a whole exome CRISPR library, and let's find all the synthetic lethal interactions that exist, and then we'll know what all the drug targets are. And in fact, there's only been a couple that have come out of that clearly. The reason is this, so that it's exactly this. If you look across all 1,000 cell lines, you find very, very few things that are synthetic lethal across the whole line. If you go down into a subset of very well-defined, say, now I'll pick out another ARID1A subset of ovarian cancer patients. Now you can find, we published this last year, we showed this last year at a poster in AACR, that in ovarian cancer, Eglin-1 is an ARID1A synthetic lethal pair, but it's not true in other ARID1A mutations in other histologies. So we know this is true, and it really is about identifying those subcontexts of patients where you can identify a synthetic lethal target pair that are very effective.
So I guess tissue of origin does still matter. That higher level biological context does still matter. It does.
And, I didn't think it would either. When I was at GSK, we actually tried to do a trial with the GSK HER2 inhibitor, lapatinib, for HER2 mutations in other cancers. This was like back in 2005 or 2006. And it didn't work. And that's because of this.
Interesting. So you've made a couple references to ARID1A. This sounds like it has surfaced itself as a high priority project at Tango. Can you describe this lead program and what excites you about it now?
Well, yeah. So first of all, it is an interesting gene to me because it's a very commonly mutated gene in cancers. We don't actually have a lead program on that at the moment. We are doing some screens, but we at the moment haven't found anything. Those screens are early on, so that's.
Oh, okay. So the gene's interesting, but it hasn't. You don't have new chemical matter aimed at that.
Not yet. I just use it as an example because it's a good example of cancers where there's not good treatment. It's a common lesion in those cancers, and it's common across multiple histologies, but the synthetic lethal pairs are not the same across the histologies.
The other thing that occurs to me immediately is that, you can get into very, very small patient populations here. How does that factor into your thinking about which programs to advance?
Well, that's a good question. You could, but I think, honestly, there's so much out there that hasn't been done yet with tumor suppressor genes, and also something we call unmarked oncogenes, and I can come back to that in a minute, that there's still low-hanging fruit for pretty big patient populations. So whether it's something like KEAP1 mutations that are 20% of lung adenocarcinoma or something like that, I think we're not talking about tiny patient populations. We're actually talking about reasonably good-sized patient populations by most people's standards.
Well, you said at the very start, you talked about tumor suppressor, the BRCA1, couldn't really do anything about it, but now we do see a way forward. Can you describe that rationale of how does one target a tumor suppressor gene via synthetic lethality?
Well, it's simple, but it's not, right? You do the experiment I just described, which is you think about what tumor suppressor gene do you want to try and find the synthetic lethal pair for, and in what histology, and you look for those synthetic lethal pairs. Now, going back to BRCA1, the first one out there is PARP. There's also POLQ, which is the company called Repair is working on that one. We have a different target that's a BRCA1/2 synthetic lethal pair that we haven't disclosed the target on, but we're in hit to lead with that looks like another good one. So I think that, for each tumor suppressor gene, there's probably more than one synthetic lethal pair, even within a histology. And then you have to start applying the standard sort of drug discovery questions like, can you drug it? How hard is it to drug? What else are its side effects going to be? Those kinds of things.
Okay. So hit to lead, is this your lead program?
We have two programs that are in about the same stage. We haven't disclosed the context of the other one. And we're going to start two more drug discovery programs in the next 3 to 4 months. One of them is going to be in microsatellite unstable cancers where the mutations are the known common ones, MLH1, MSH2, MSH6, sorry. And the other one is a combination with a RAS inhibitor. It's a combination with a MEK inhibitor in RAS mutant tumors.
Are we talking about small molecules pretty much in every case? Okay. So the path forward here, pretty straightforward. Hit to lead optimization, in vivo tox. Have you said like anything about timelines where you think you might end up in the clinic?
Yeah, we're hoping to be filing INDs on our first two programs toward the end of 2020 and in the clinic in 2021.
Barbara, as I was getting ready to talk to you, I do what reporters do, which is we look at the company website, and I was really struck, because you've got this kind of background, which I alluded to earlier, that it could have led you to be in one of these big corner offices with wood paneling and walking around with a tweed jacket or something, and there you are. You are on the website, and there's this whole team of people who look like they're having a lot of fun. They're all wearing white shirts and I think purple tennis shoes, and it's very startup-y. What's the story there?
I love it. Well, the pictures in the white shirts, I was actually the day we moved from Hurley Street, which was where we were for the first 18 months or so, subleasing from Editas, into our new space at 100 Binney. So we sort of, as a sort of company event, told everybody wear white shirts and jeans on Monday. And then we also had a contest to design the official shoe of Tango Therapeutics, and the winner was selected and made. So that the shoes everybody has on are the Tango sneakers from the Tango sneaker contest.
Well, the purple, it all seems very consistent. That's part of your logo in your brand, I guess, so to speak. But no, I mean, I think there could be some silliness here, but I think there's something interesting going on in a boom time like we're in. I mean, when I would have looked at a company's website at your stage of development, say 5 years ago, I think the website would have been more geared toward investors, as in, look at how attractive we are for investment. And when I look at your website today, it looks to me more like it's geared toward employees. Like, look at what a great team we've got here and why you'd want to come work with us.
Oh, that's an interesting thing. When we built the website, and I personally was pretty involved with that, what was important to us was that it reflect us as a company and as a team. And what I think is my own personal philosophy, but I think it, as a result, probably permeates the company, which is that really good science and really important drugs will speak for themselves, right? That what we want to do is explain to people what we do and do a good job of it, and that in itself is how we want to be seen, right? We're good scientists doing really important things for patients on a timeline that makes sense for investors. But it's about what we do.
Okay, so what you're saying is,
doing it.
So it isn't like this was intentional, like that you built the website with that recruiting kind of mindset in mind.
Actually, it's funny. I didn't really think of it until you said it. You're right. Who are the people that are going to look at this? People who want to work here are going to look at it, so that's that page. People who want to potentially partner with us from pharma or invest in us are going to want to understand a little bit about the science, so that's those pages, and they want to know who we are, so those are those pages. But the feel of it, I hope, just reflects who we are.
Well, certainly you're always going to have multiple audiences in mind when you build a company website. I get that. But it just, I've seen this with other companies too, where I think, we're in a time when there's a lot of money out there that wants to invest in biotech, a tremendous amount of cool science to invest in. But when I talk to the VCs, I hear people, fret a little bit about, talent being hard to come by, especially the kind that can run companies.
That's true. And I didn't in any way mean to imply that that's not a big focus for us, that we work hard to recruit and retain the best talent. And the website can be a tool for that. So we wanted it to reflect who we were. But yeah, the website wasn't built with that express purpose or slant in mind.
So what are some of these big learnings for you in startup land, in building a company? I'm guessing you didn't, have a course on this in graduate school.
I didn't, but I learned a lot at Third Rock, right? I spent time, like a very intense time for almost 3 years, both building a couple of companies with people who really do it well and watching a bunch of other companies getting built during that time. So I think that I had the luxury that a lot of first-time CEOs don't have. And I certainly have friends around the world that are doing this now because it's I think now much more common to find people from big pharma that have moved into biotech roles. I just think that in a way I did have a course in this by spending that time that I spent at Third Rock. What are my biggest learnings from here? I know this sounds a little operational, but I would say so far my biggest learnings are that you have to it's different than building. Let me say it this way. It's different than making an organization that's already exists bigger, right? That's what I did at Novartis. I had an organization, it had a structure. It grew from, 120 people to 400 people by the time I left. But the structure and what everybody was doing was largely fixed over time. The projects rolled through, but the organization was the same. In a startup, the organization has to morph, and it has to morph pretty quickly over time. As you start to roll a pipeline out that didn't exist, right? So you're constantly adding on new functions, and you kind of have to think about what's the right structure and how to manage that when there's 11 people as opposed to when there's 80 people. So I think just the dynamics of truly building a company as opposed to making an existing organization bigger are different, and I hadn't really thought through that.
Well, a lot of those structures that you allude to were already in place, put there in many cases years ago, often by a predecessor who may or may not even be at the company. Maybe people don't even know exactly why they set it up this way, but it's sort of like, this is always the way we've done it, so we're just going to keep doing it that way. You see that a lot.
Sure, but it also made sense, right? There's a clinical group, there's a clinical operations group, there's a pharmacology group, there's a statistics group. That was all made because now, of course, at a big company, there's a pipeline that rolls through that, whereas here, we're both building the organization and building the pipeline at the same time. So it has sort of different implications. On the very positive side, I would say one of the things I love about this is, for me, it's kind of the perfect mix of what I've done in the past, which is that it has all the sort of cool, cutting-edge science of being in academics without the headaches. And it has all the pragmatism of saying the reason we're getting up and doing this every morning is to do something important for patients of big pharma without the headaches of that. And it's all in one place, right? I spent a lot of time flying all over the world for GSK and Novartis, but now I'm sitting in my office looking through my glass doors, at the entire company. And I can just go talk to anybody I want to in 2 seconds, which I love.
Is that one of the ways you like to manage, just walking around and talking to people, see what they're doing?
Yeah, absolutely.
Yeah, so you feel like you're close to the science and pursuing interesting questions. All the while, hopefully getting from point A to B to C.
Absolutely. And I also love it because it's just a spectacular team. I mean, I trust everybody out there to do their job better than I could do it, right? So, for me, it's just a dream situation.
Where do you think this will go? I mean, you're not the only company working on synthetic lethal combos. And whenever, there's like a good idea in pharma, a lot of people get organized very quickly to pursue it. How do you see this field or this kind of subsector within oncology evolving over the next few years?
Yeah, I think that's absolutely right. And I think even going back to CRISPR, it's almost like eventually it'll be almost like PCR. It's becoming a basic research tool that's so versatile that it's hard, even all the validation we do now the target validation work really depends heavily on CRISPR. I hope that does happen. I would say we've talked to a lot of pharma companies over the last 18 months, and I would say most of them are not looking to build this kind of a platform internally. They're looking to partner outside with other companies that are doing it. And there's different ways to do it, and there's different contexts. So, KSQ and Tango are taking pretty different approaches and will, I'm pretty sure, end up in very different target spaces. In the end, it'll only be good for patients if this is what happens and there's multiple new important drugs that come out of it. Now, for Tango as a company, I'm excited about the fact that we're one of the first, and I think we really are good at it. And so I think that will hopefully be an advantage for us as a company. But in the end, it still ends up always being about why we do this. And I don't want somebody else to not do it.
Well, there's a Versant-backed company, I believe, in synthetic lethal discovery. I don't know if they're using a similar kind of platform for discovery as you guys, but back to KSQ, what would you say would be the big difference between your approach and theirs?
What I understand about KSQ, which is only what's in the public domain, is that first of all, they're using a different kind of CRISPR approach. They use CRISPR-I and we use CRISPR cutting that may get you at different targets. They also have a heavy focus on in vivo screening for T-cell targets for immuno-oncology. So again, likely to end up in very different target space, but we don't know a lot about what they're doing.
Well, there's definitely a market for anything that can assist T-cell therapies or the PD-1, the checkpoints, which is different from what you're trying to do.
Yes, but I do believe that this is really a huge target space, and that I believe that we are getting close to, and actually soon will have more targets than we know what to do with. And I think that's why I go back to saying that it's just a big target space, and as people get good at this and understand how to do it and how to get at the combinations, and this is another important point, right? That it's going to take combinations of drugs to cure solid tumors. Whether people like it or not, we don't get to decide that. It's just a fact. And I think thinking that there's any single agent therapy out there for solid tumors is probably pretty naive. So even if we find the first synthetic lethal drugs or the first couple of synthetic lethal drugs are among those discovering the first couple, there's many more. And then there's how to combine them and what are the right combinations. There's this whole space, what I call unmarked oncogenes, that we haven't even talked about. That's an example of important nodes in specific genetic contexts that aren't marked by mutation or amplification, so you can't find them by sequencing. That's an example like the CDK4/6 inhibitors in ER-positive breast cancer. Neither estrogen receptor nor CDK4/6 are mutated. It was a hypothesis that that would be a good combination, but you can't find every combination in all cancers with hypothesis testing. So there's just a ton of stuff that's still out there to be done here.
Yeah, for a long, long time, it's been about what's the one target and the one drug for that target. And now it seems like we're in there's still a place for drugs like that, Herceptin being an obvious poster child for it. But, we also at a very high conceptual level could be going toward some more combination therapies. And a little bit like the antiviral story with HIV and hepatitis C, multiple drugs.
Every cancer that is cured, that is curable in humans, and there are a number of them, are all cured by combination therapy, right? Whether it's Hodgkin's disease or testicular cancer or childhood leukemia require multiple combinations. And that goes way back to what Bob Weinberg predicted 40 years ago, that every cancer has somewhere between 3 and 6 important genetic lesions that need to be targeted to kill it.
Well, I guess we sometimes forget about the combinations because it's sort of like standard backbone chemo along with the new thing.
And remember, chemo is even combinations. All effective chemotherapy for solid tumors is curative chemo is combinations. And so cancers are just too plastic and too mutated to ever think that we can get at them with single agents. CML is a completely different thing. CML has a single genetic lesion, and that's why a single drug can be curative in that setting. There's no solid tumors that look like that in humans.
Yes. We're all well aware that people tried to replicate the Gleevec story in other tumors, and it's just, if only it were that easy. Barbara, it's been a great pleasure having you on the show today. Thanks for joining me on The Long Run.
Thanks so much. Thanks for asking me.
Thanks for listening to The Long Run. a production of Timmerman Report. Pedro Rosado of Headstepper Media was the producer and editor. Music comes from D.A. Wallach. And thanks for listening. If you're interested in sponsoring the show and getting your name out in front of biotech thought leaders, send me an email at luke.timmerman@protonmail.com. See you next episode.